Welcome and good morning to our today's lesson. I'm Meshach Kodiko. So today we're going to talk about the practical applications of genetics. We all know what genetics is. That is the study of inheritance and variation, why there are similarities and differences among living organisms. And then we want to now look at these differences and similarities. How can we apply them in our day-to-day life so that we come up with ideas on genetics. So you can be given a question like, describe briefly the practical application of genetics. So we have quite a number of areas on which we can apply genetics. To begin with, we have to look at what we call blood transfusion. Blood transfusion is the transfer of blood from a donor to the bloodstream of a recipient. And the blood can only be transferred when the blood is said to be compatible. That is, compatibility here means that we don't have agglutination, or rather coming together of the red blood cells. Now, blood groups are genetically determined, and we know that we have 4 blood groups. That is blood group A, B, O. There are 3 blood groups there, though we can talk about 4 because we have the AB. So is this ABO blood group that is determined by what we call multiple alleles that occur in groups. So a person of blood group A can only get blood from another person with blood group A. Why is that the case? Because we know very well that if you are having blood group A, you have antigen A but antibody B. So another person with blood group B will have antigen B, antibody A. Then one with blood group AB will have both antigen A and B, but will lack antibody. But a person with blood group O, we know better that O is recessive, it cannot express itself. So blood group O does not have antigens, but has got antibody A and B. So maybe we want to look at why is it that a person with blood group A can only get blood from another person with blood group A. This is because we don't want, during blood transfusion, we don't expect agglutination to occur. That is, a person with blood group A has got antigen A, but antibody B. So this person, you cannot give the blood to a person with blood group B. Why? Because this person has got antibody A. Whenever the blood group corresponds to the antibody, then there is going to be agglutination. So to prevent agglutination, a person with blood group A can only get blood from another person with blood group A, but because that person is having what? Blood group, antibody B. That means that there is not going to be correspondence in the blood group and antibodies. Same to the people with the blood group B can only get from blood group B because they have antibody A. So we are saying that the person with blood group A can only get blood from blood group A so that that agglutination does not occur. That is the clumping together. Then genetics determines the dominant dominant and recessive genes as well as the antigens. We also have the antibodies and the rhesus factor. It is through genetics that we are able to understand what is the antigen, what is the antibody, and what does the rhesus factor mean. And is it dominant or recessive? That is through blood transfusion. Number 2, we have what we call plant and animal breeding. Due to increased food shortage, farmers have been advised, or rather have been taught on how to blend these characteristics so that they come up with fast growing plants and at the same time animals that are bred so that good qualities are reached within the shortest time possible. So genetics is applied in order to produce varieties of crops that are most suitable to the needs of the man. That is because of the increased food shortage. So we need to get varieties that are able to grow fast and provide food for the fast growing population. So in this case, it is through genetics that we are able to determine the varieties that are able to grow fast and after growing fast, we do that in terms of what we are calling artificial selection. We select those varieties with desired qualities, after which we are going to produce them. That is the first growing, those that are resistant to diseases, and maturing within the shortest time possible. Then we have what we call genetic counseling. Sometimes we've had a lot of diseases that are disturbing couples in their families. Some of those diseases are brought about by what we call heredity. Heredity here is the ability of a characteristic to be transferred from one organism to another. So it involves advising people about hereditary diseases so that they can make informed decisions. For example, one that we know is the hemolytic disease of the newborn. Somebody may be asking why he saw children are dying, the first children are dying. That could be because of the hemolytic disease of the newborn, and that can only be realized, or maybe correct information can be got through genetic counseling, so that you are told how the characteristics were able to be transferred from the parents to the offspring, or even the formation of these disorders there. Then we have what we call genetic engineering. By definition, genetic engineering is the manipulation. You manipulate, you alter, separate, and then you transfer a desired quality from an organism which is high quality to another one which is low producing. So it is a technology that involves the manipulation of genotypes of an organism with the desired traits, or rather the qualities or characteristics. Then the genes are then transferred. Coding. When I talk about coding here, coding means that it copies the same information from the original template. So the genes are transferred, coding for the desired trait from one organism to another. So the organism onto which the desired qualities are taken is what we are calling the recipient organism. So when that happens, then we are going to have organism with improved production, what we are calling hybrid vigor, or rather heterosis. So that is genetic engineering. Another thing that we have to look at is crime detection. We've had a lot of problems in detecting those people who have committed crime, but from the information of genetics about the fingerprints. Let's say, for example, somebody uses a gun to kill the other one, then we can use that gun to go scan so that we look at the fingerprints, look at it versus the suspect, so that we look at the pattern of the fingerprints that are there, knowing very well that fingerprint is an example of a discontinuous variation. That means that each and everyone in the world has got a different type of fingerprint, so you can easily be determined. Or even those people who have carried out crimes like rape, we can also use the semen to determine the DNA of the person, and then we get the individual who has committed that crime. Then we also have what we are calling cloning. Cloning is always done in plants, and this involves making identical. When I talk about identical, meaning the same copies of the gene or the DNA. It is used in plants, that is the tissue culture, like the one that is carried out in bananas, so that we develop those plantlets which are fast growing, and then again they are resistant to diseases, and so many plantlets can be produced within the shortest time possible. So that is cloning. Last but not least, we are talking about gene therapy. Gene therapy here involves injecting genes into patients of a certain disease. That means that you identify a gene, then you inject it to another organism. The injected genes alter. Then they are going to manipulate the metabolism or the working mechanism of the body of that disease, and therefore it brings about a cure of that disease. So we have now seen the 7 practical applications of genetics. We have talked about blood transfusion that we now understand. We've talked about plant and animal breeding, which can also be applied there by production of desired qualities that are fast producing. We also have what we are calling genetic counseling, which we have seen on the diseases that are there, that maybe you can be advised on what to do about these diseases. Then we have talked about genetic engineering, where we alter or rather manipulate a characteristic and then transfer to organism that is lowly producing. we've talked about crime detection based on the continuous characteristic, discontinuous characteristics of the individuals. Then we have seen cloning, where identical copies of a characteristic is made so that they are then transferred, and then there will be fast development. Then lastly, we have talked about injection, the gene therapy, where the genes are injected into the metabolic system of the patient, and therefore to bring about cure of that individual. Thank you for listening to our lesson of today, and let us meet next time.